a very interesting story to tell. So before I introduce her, how many here think that we need better alignment between clinical research and clinical care? Everybody, right? Okay. Well, exactly, right? So that's a noble goal that is actually very hard to operationalize. And today we'll hear, I think, hopefully some of the wonderful work that Amy's doing with the multiple hats that she wears. She's a professor of medicine at the Duke University School of Medicine, director of the Duke Center for Learning Healthcare here at the DCRI, as well as the director of the Duke Cancer Care Research Program at the Duke Cancer Institute. She's got almost 400 publications, is an internationally recognized expert in health services research, comparative effectiveness, patient-reported outcomes, clinical informatics, and patient-centered care around end-of-life care, oncology, and a variety of other growing areas. She's also, in addition to her role here at Duke, the Chief Medical Officer for Flatiron Health. And this is a health technology company focused on organizing the world's cancer data and making it useful for patients, clinicians, researchers, payers, and policymakers. She participates in a number of high-level organizations that seek to help spread learning healthcare systems and learning healthcare system principles and architecture in order to allow all of us to hopefully identify ways to align clinical care and research better. So please join me in welcoming Amy here today.
Thank you, guys. We were laughing as I came in, so I wore my Duke blue. So this is the CME slide for those of you ready for CME. And what I'd like to do today is talk a little bit about where we're going in terms of creating infrastructure for learning health systems, thinking about it from a health information technology perspective, and really kind of walk through the problem statement as to why, and then at least how we're trying to solve this at Flatiron and what that has meant happening within the context of what's happening. This is my funding disclosure slide, and one of the reasons that I like to put this slide up, besides the fact that I have to, is because I think it reminds me of the importance of all sides of healthcare coming together to solve these problems. If we want clinical care and clinical research to be better approximated, we actually have to align incentives for all parts of the healthcare system. And this includes manufacturers and industry, it includes healthcare systems and healthcare providers, it includes researchers, patients, payers, policymakers, and certainly clinicians. So RJ is here, and RJ has helped to remind me through pictures many of the important things about healthcare, and one of them is that we're all turtles. 14 to 17 years to get from discovery and new evidence development to widespread clinical implementation. And if you think about that from the perspective of being a patient, that's absolutely absurd, right? Why should it take 14 to 17 years for us to go from what we know works to doing so in widespread practice? And I always anchor myself to a patient that I met in 2009 named Janet. Many of you have met Janet in different ways, shapes, and forms over the years. Sometimes she even has a different name, which is Sarah. But she really fundamentally is really the person who helps to remind me why I do this work and how to think about the perspective of the people that makes the most difference. So Janet is 37. She's an emergency room nurse. I see Krista in the back. Krista was the person who first took care of her. In fact, you can kind of imagine Janet, because she's got this curly red hair and freckles on her nose, and she is a woman with a 3B melanoma, where at that time I called it the 50-50 disease. When she came to see me, the issue was that the surgeons had completely removed the melanoma from her arm and resected the tumor from underneath her arm. But in fact, even though the disease was gone, she was at a very high risk, 50% of the disease coming back and taking her life in 5 years. And the question that we had that particular day in 2009, which is exactly the same question as we would have today in 2015, is whether or not we should give a woman like Janet adjuvant treatment to reduce the chance that the disease comes back in the future. In 2009, scarily, the same treatments are the same ones that we have in 2015. The question was around adjuvant interferon. a drug that potentially could reduce the chance of the disease coming back, but we weren't sure. The only thing we were sure about was it would make you feel really miserable when you received it, reduce your quality of life, and make it very hard for her to do the work that she cared about. She was a woman who was taking prenatal vitamins when I got to see her. She wanted to know whether or not interferon might impact her fertility. I had no information, no data. And in 2009, same with 2015, we could predict her risk of death. 3B melanoma gave her a risk of about 50% at 5 years. It may be better in 2015 with new drugs that are coming out, but it's still pretty awful. She had pain underneath her arm, and I had tools in my toolbox to treat her pain. But I actually couldn't tell her, Janet, for you, with your red hair and a mother who died of melanoma and your particular genetic predisposition, here's what we should do based on any real evidence. I didn't know her risk of infertility. I could tell her that interferon would probably reduce her quality of life, but I didn't know in what ways. Was it going to make her depressed? Was it going to make her so fatigued? Was she going to have other problems? I didn't know. And I didn't know how to talk to her about the impact of her mother's heritage in Janet's current life, or for her family that she was thinking about for the future. And so, I've always used that as the anchor, that how can it be that sitting in clinic we have so many unknowns and so few knowns about taking care of the patient sitting in front of us. When we think about cancer, often we imagine that the cancer space is the place where we have more data and more activity focused on making decisions based on evidence in clinical trials than anywhere else, but it turns out that 97% of our patients in cancer are not represented in our clinical trials. And the world typically looks like this. We have lots of activity going on in drug development. In fact, we have fewer and fewer phase 3 trials in cancer these days. We have accelerated approval programs, lots of drugs coming out based on phase 2 data. The drug gets licensed, and then we move into the post-marketing space where the majority of care is not collected, studied, or understood in any systematic fashion. And so what happens is for Janet, her story does not enter that overall system of data. And similarly, all of the people receiving care who also had 3B melanomas don't inform how to take care of Janet either. So we have this fundamental problem that the majority of the care that we're providing does not enter into the overall world of evidence development and inform the stories. Late 2000s, 2015, we've seen a whole bunch of conversations happening at the same time. Personalized medicine, patient-centered care, healthcare delivery, healthcare quality, comparative effectiveness research. And often you'll look at a person's CV or background and they'll say, I'm a comparative effectiveness research person, or I do personalized medicine. And even here in the DCRI, we often think about the people who do those different kinds of work as fundamentally different phenotypes. And yet, in fact, all of these different co-occurring ways of trying to develop evidence and improve healthcare have at their baseline linked patient-level data. And if we actually could get to a system of linked data organized in a way that we can make sense of it at scale and with speed, we can start to solve all of these problems simultaneously. And so, with that backdrop, that has been a fundamental underpinning of this conversation of learning healthcare systems. Cancer. So trying to bring all of those organized data points together in cancer isn't a 2015 conversation. It isn't even a 2005 conversation. In fact, we've been working on trying to do that in some systematic way across the overall cancer spectrum since the mid-1970s. We've had the tumor registry system, the clinical research community, and the cooperative group systems. We've moved towards conversations around data warehousing, like the data warehouse here at Duke, the DSR. There have been many groups for 40 years trying to solve this problem. But it's been really, really hard. And we still are in a place where 97% of our patients aren't represented, except for within the tumor registries. So why has it been so hard? 2009 or so, this was the proposal about creating a national infrastructure to start to support learning healthcare systems. We had the conversation around meaningful use, and maybe meaningful use was going to be our way of getting to an overall system of liquid data that we could make sense of and speed up new knowledge generation. But it sort of feels like we continue to be stuck. So why is that? We said, the electronic health record, that's going to be the answer, right? Yes, if we put $1 billion down or $100 million down or something else and make sure that our healthcare system has an electronic health record that we can count on, of course we can handshake with UNC or University of Michigan because we have the same systems and be able to move data from one point to another. But in fact, it turns out that the electronic health record has not proven to be the mighty force of data liquidity that we expected it to be. And so actually it feels like accessing data through the electronic health record sometimes feels a bit like this. You can't get to it. And we saw in the New York Times in September that doctors are having a hard time sharing. At HIMSS a couple weeks ago, interoperability was all the conversation. In fact, at that point, interoperability is now not meaning can we share data, but simply can we get one system to start talking to another system, just even to do basic applications. So we've got a long way to go till we can actually start sharing individual linked patient level data. And so the problem looks like this. The system is highly fragmented. You got Epic at 3 different institutions, all of whom are interested in talking to each other, but they still can't talk to each other. And that's when you're all in the same electronic health record. It gets fragmented further when there's Epic and Cerner and Athena Health, and we've got 5 different electronic health records in oncology. So we've got Altos, now OncoEMR. We've got Inomed from McKesson. We've got 5 different ones, including Epic. Secondly, there's this other problem, which is that we all know that if we want information that we can rely on and analyze, it actually has to be in digital format. It's got to be something that you can calculate or sum or count. And much of the information that really matters lives in our notes, in the radiology report. And so, in fact, a second huge problem with getting to the digital infrastructure that we were all hoping for to solve this is that the data live in 2 different places. Structured data, which is the digital stuff, and unstructured data, the PDFs. And all of that has to come together. And actually, the PDF problem is even worse in oncology because basic things like stage and biomarkers typically aren't in any digital feed. The third problem that makes this so hard is that all of the information is living in these repositories right now that are fundamentally data repositories but aren't actually really about pulling data out and moving it around. Electronic health records, for the most part, are dumb repositories. And so data extraction and putting it together in some kind of meaningful way against one common data model is a holy grail we have a hard time figuring out how to deal with. So, it's hard. Janet reminds us there's promise. If we could get all the data organized in a way that we could make sense of it, we could solve a whole bunch of different problems from precision medicine to healthcare optimization, but how are we going to do it? So one of the things that we worked on in the 2000s here at Duke was trying to think about how might you solve this problem differently. And we spent a lot of time trying to think about what if the real solution was through the back end. We wrote a Go grant in 2009, and basically one huge section of that grant was around this concept of getting there through the back end. And essentially, one way that we proposed, even in 2009, to start to get there was by dealing with the core components of the problem in a systematic way. The first was to say, listen, let's get to the back end of the electronic health records. In 2009, we were just talking about trying to get to the back end of Duke's system, and we were actually trying to figure out, like, could you do Duke and UNC? But what if you could get to the back end of all the systems across a discipline like cancer? Second, dealing with both the ability to figure out what's in the structured data as well as in this unstructured data and put it together. And third, mapping everything to one common data model so that data are already pre-organized in a way that you can analyze it and that those analyses can come fast and you don't have to wait. So that was the proposal that we had in 2009. And now the question is, can we really pull it off at scale? So what I'm going to do now for the next 10 minutes or so is tell you a little bit about what we've been doing at Flatiron, which the goal is to really introduce to how we've been moving this forward within one company. Importantly, I work there, and in terms of conflicts of interest, I think what we do is pretty cool. But there are other companies out there trying to solve this kind of problem in other ways, and we can talk about that later if you're interested. So the first thing that we realized was, and it goes back to when we were trying to write our PCORNet grant, and how do you actually create partnerships across something like PCORNet? The partnerships with providers is key. And you have to actually create alignment and of incentives so the providers actually want to be part of an overarching network, and where sharing makes sense for the providers, not because it's some kind of altruistic set of activities, but because it's good for their clinical practice and the care of the patient sitting in front of them. And so partnership becomes the number one thing. Often I find researchers who say, but my job is research. My job's not to make the clinicians happy. And one of the things we talk about a lot in the Center for Learning Healthcare is that in order to do the research, first we focus on the clinicians and the patients, and the research then comes as a byproduct. And as I'll show you in a second, it's been the same premise inside of Flatiron. So at Flatiron, we have two products. It took me a long time to get product speak, but essentially these are the two ways that we deliver our solutions to healthcare providers. One is on the right, which is OncoEMR, which is the largest cloud-based electronic health record in the country, and so for oncology. And so there are 5 oncology EHRs, 2 of which are cloud-based. And the advantage of being a cloud-based EHR means that the electronic health record, by definition, is on one common data model. The whole system doesn't get tweaked and tuned for each individual practice, but rather all practices do things in generally the same way. Right now, as I'll show you, probably about 1 in 5 cancer patients are cared for by oncologists who are on that electronic health record. However, not every cancer provider in the United States is using that oncology EHR. So you also have to have a mechanism for providers to also map to the same data model if they're actually using a different electronic health record. Epic is a big one right now in oncology. So is Inamed. These all then are data come out of the back end, and these providers have access to a different kind of tool that we call OncoAnalytics, which is essentially a tool that provides population health solutions. And in order for those providers to get access to the population health tool, the data have to come out of the back end of their electronic health record, be processed and scrubbed, as I'll show you, and then given back to the providers. And so basically by having these two tools, OncoAnalytics, which predominantly is for big practices and for academic centers, and OncoEMR, which is predominantly a community-based electronic health record, we now have a mechanism through which data for about 20% of cancer patients come and get mapped to one common data model. So this is the network. It's actually a little bit old, but one of the things to sort of point out then is you're able to create geographic representation really fast. And if we sort of look across this, the other thing that you see here is this is 2013, 2014. We only were using OncoAnalytics as the platform at that time, really trying to figure out how do you pull data out of the different electronic health records in oncology. And this is when we realized, hey, it might be really smart to buy our own electronic health record and actually own one of those systems so that we have the tool suite to do this at scale. So then the next thing, once you have partners, a network of providers all essentially able to share and work together across a network, the next thing you have to do is actually have a way of processing the data that's coming in so that you can hand it back to them in some way that makes sense. And the data processing becomes sort of the main thing that we focus on, and lots of people always want to know how is that we do this. Is it natural language processing? Do you have a bunch of fancy algorithms? What does it look like? And so I thought what I'd do is sort of invite you behind the curtain for a moment and show you what we actually do. So the majority of the data come in through what we call the structured data pipe. I kind of like always imagine that I'm in Canada and there's the big pipelines kind of coming through North America, and this is the big pipe of data. that comes from all of these different kinds of providers. Some providers have a single EMR. Some providers like Penn, have all kinds of systems, and finding the data across Epic and Clarity and all the different places that we have to pull it in from means you essentially are trying to pull from their mega pipe into an even bigger mega pipe and map to one common data model. And this is sort of an example of normalizing the structured data. And the funny thing is, this is the example we use at Flatiron. It's an example from albumin, and it's the same example we used at Duke in our Go grant in 2009. Ironically, it just happens to work really well and be pretty obvious. So this is albumin from one practice in Florida. And what you see here is that the things that are in darker gray are the albumins that represent serum albumin. That would be the true data that you would want to map to a common concept of albumin. And so the first thing you have to do is weed, kind of go through all the different weeds of the different kinds of albumin, get rid of the urine albumins and the CS albumins and microalbumins. You have to deal with the fact that many of these laboratory tests, some of them are homegrown, they come from LabCorp and Quest. Sometimes people type in the name of the albumin incorrectly. So you actually have to ultimately normalize like this and harmonize across every single one of these practices and every single one of the lab tests and other structured feeds to ultimately get to one common concept and mapped to a common value. And so that kind of structured data processing happens across the entire network and all of those practices, and that's basically, we have a whole bunch of people that spend a lot of time doing that. But the real value of data in oncology and getting things to a place where all of the data points that are historically missing start getting filled in come from the unstructured data, the PDFs, the stuff that you really wanted in the first place, like whether or not she's got bone mets. And in order to do that, the solution looks a little bit like Google Maps. So if you think about Google Maps, like one of the first times I noticed that they could see my front door, I was a little freaked out. Right? I was like, how is it that, essentially, I thought the whole thing was done by, some satellites and fancy cameras and, basically some software that put the story together. And lo and behold, I was in San Francisco one day and I saw a car like this and realized that, in fact, they've got people driving cars up and down the roads, and then technology that integrates the information feeds from the satellites and from the cars and ultimately puts the whole picture together to give you a map and Google Earth and your front door. And so we do something quite similar. What we did was we started off with the concept of good old-fashioned chart abstraction. And I know Danielle is here, and she sort of was like the queen of our chart abstraction for a very long time. And inside of our team here at the DCRI, we often might do a chart abstraction project where we have a 30-page CRF. We have essentially Danielle and colleagues, pick up the chart or maybe get on the electronic version and start filling out all the information in the CRF. Hopefully we've got some good quality standards around what we're doing and some kind of review, but it takes a long time, it's inaccurate, it's expensive. The first thing that we did at Flatiron is we say, okay, guess what? When Danielle is looking for biomarker data, she probably doesn't need to be looking in the radiology report, right? So let's start by breaking down all the different kinds of documents that live in an electronic health record and categorize them so that we think about what documents are relevant for what tasks. The second thing we said was, wait a second. Going from top to bottom on a CRF doesn't make a lot of sense. So first of all, there's that emotional angst that goes along when you look at the top of the form, you think, oh my God, I got to fill that whole thing out. But further, there's a lot of context switching that happens in your mind. First, you're doing stage, then you might be doing demographics, then you're spending a little time looking for progression events, and then, by the way, you're looking for biomarkers over here, and now you got to understand what all those mutations mean, right? But it turns out that if you could actually break this into different tasks, some people are really good at understanding the mutations, some people are really good at doing stage, etc. So what you do is you modularize it. You package up the kinds of documents that the person who's doing stage assessment for cancer stage should be looking at, and you hand them to the person who's most expert at identifying stage. And you have them do just that task. And similarly, you do the same thing for biomarkers. and for assessment of tumor response and progression. And then you build into your software not natural language processing and things to take away the people skills, but actually continuous feedback loops to figure out how do you teach all of the steps in the system to get smarter and smarter and smarter. So in fact, what you find out is that it turns out that the people in Colorado, for whatever reason, are really good at stage. So you're going to start matching as much of the work for staging to the abstractors and the kinds of technical people who live in Colorado. And meanwhile, some folks in Boston, particularly good at biomarker assessment, and it turns out that certain kinds of trainees, so people who are certified tumor registrars, do some kinds of things better than nurses. And you start putting all that together. And so what you end up with then is a modularized system where things have been broken down into tasks and a continuous feedback loop of learning to try and get them smarter and smarter and smarter. And further, you actually can parallelize the process and test it even further. If I don't like the way the data are coming back around a particular variable, I'll send it out to 7 different abstractors. I'll adjudicate at scale and see what happens. And start using a series of tests to figure out what if we tweak the way the question is asked and send it out to the same 7 again? What if we need to understand, is this a sensitizing mutation or not? And we ask the word sensitizing in 3 different ways to try and get to the point where the abstractors really understand what sensitizing means, et cetera, and start to optimize more and more. Only once the system is optimized do we start adding in more algorithms. So it's true, we have some algorithms that start to use natural language processing to find really basic things, like insurance status, because it turns out insurance status is often not in a structured feed. But it also turns out that we can get much higher quality and test the quality of the variables very, very carefully when we have human abstraction done at scale. And so really we call this technology-enabled abstraction. And so one of the things then that does is help us fill in those data points that are historically hidden in the unstructured data. Finally, the last piece is that by pulling everything out of the back end, by mapping it, and by pulling out the unstructured data points, everything gets mapped to one common data model. The data are organized in a way that they're highly ready for analysis at all times, and that all of the analytic algorithms that we write don't need to be changed. We can actually now set up the initial algorithms ready to go, and now start tweaking the final question. So what does that look like? The first thing that we do is we start working on making sure that we can understand longitudinally the complete patient story. One of the things in learning health systems is recognition, the importance of longitudinality. Patients live across time, and their data aggregate across time. And so we start to build these maps that show all of the different kinds of data feeds and the data points coming in across different data types so that we understand how much information we have around individual patients and the relationship of that information to each other. And Arif is smiling, and one of the reasons I think he's smiling is because we've been seeing this picture inside CLHC since 2009 or so, when Susan Locke first started drawing them. And in fact, we drew exactly the same pictures at Flatiron. The next thing we do is we spend a lot of time understanding the ability to complete variables and solve the problem of data missingness. So in essence, if we just look at structured data, and here are classic data points in melanoma, what you see here is that metastatic diagnosis, so understanding the patient has metastatic disease, increases from 35% to near 100% when we fill in that variable using unstructured data processing. And it seems kind of crazy that something so basic as whether or not this person has metastatic disease would be missing from the electronic health record in any structured data feed. But in fact, as you can see, 65% or so of the time, that's the case. The date of the metastatic diagnosis, BRAF status for a biomarker, for example, whether or not when the BRAF status was actually tested and what the value was. And interestingly here, we can more commonly see that the BRAF was tested because that might have gotten a specific code to go along with the cost, but it's very hard to actually get the result without going directly into a PDF document for BRAF. We can integrate the patient's data, that longitudinality, with other data sources. As I'll show you in a second, we're actually starting to integrate our data with foundation medicine, so next generation sequencing. Claims data is something that we spend a lot of time integrating with. So we start to integrate individual data sets at the patient level so that we start to have more and more information about the overall patient. Now that we have the complete patient story and we've filled in missing variables, the next thing we want to do is get to pure cohorts. Cohorts of individuals where we understand who we've selected in and how we're going to monitor them across time. And this part, Michaela, is just for you. And the bottom is the old-fashioned way of doing this. So if we take the Flatiron data set and we apply the usual case filter for melanoma using an IC9 code, which would be 172.x, would be the normal way we would do this. And then when we're trying to identify, for example, patients with metastatic melanoma, and I was talking to Jeff Cloud just a little while ago, and Jeff was working on different kinds of patients using the Medicare data set. Typically what we would do is if we need to find patients with metastatic disease, we'd add a metastatic code. So 196, 197, 198 would be our ways of finding patients with a MET code. So you'd end up with melanoma plus a MET code hopefully gets you people with melanoma. And here, if I use an IC9-based approach, which is what we would normally do, I get 459 people with metastatic melanoma. How come it is then that I can tell you that a technology-enabled abstraction approach from the same data set gets me 813 people with metastatic melanoma? Well, it's because of this. So first thing we do is we do use the melanoma code to find those patients because we know that those IC9 codes are pretty good. But then we go to the chart and we look at the pathology and confirm that this particular person indeed had melanoma. And we also do something else. We say, yes, we're only looking for skin melanomas. Because clinically, we know ocular melanoma and skin melanoma are two different things. If it's underneath your fingernails, between your toes, on your hands, those melanomas are different, and they actually don't belong in the same cohort. So we get rid of the kinds of melanomas that don't belong in the cohort that you couldn't have found using an IC9-based approach only. Then we also confirm that this patient has a metastatic focus. As I showed you before, a metastatic focus is only present in about 35% of the time in structured data feed. Why is that? Because there's actually not a reason financially to code it. And when it does get coded financially, it's because I'm giving, for example, a bone agent to try and protect the bones. And when do I do that? I do it in, for example, prostate cancer. So it turns out that a lot of these guys down here have a MET code that wasn't from their melanoma, but was from their prostate cancer, which is co-occurring, and that those are two different things. And Michaela's shaking her head because this is the problem we run into. So if we do an overlap, this is what we see when we start to build these cohorts. If we use an ICD-9-based approach only, 151 people would have been wrongly identified as having metastatic melanoma, when most frequently what they have is metastatic prostate cancer or metastatic breast cancer and a small, thin melanoma, and melanoma was not their driving disease. Or they will sometimes have, it's rare, ocular melanomas, palmar, plantar, etc. And that 505 people would have been not put into the cohort based on our secondary, based on our IC9 codes, but have been found using unstructured data processing because, in fact, these are the people who had metastatic disease where there was no metastatic code to begin with. Now we can actually get the data set ready for all kinds of analyses from that point forward. I'm just going to show you a couple of things that we've been doing over the last 2 months or so. So this is that same melanoma cohort. We currently grow our cohorts right now inside of Flatiron, kind of at a rate of, we increase them by size by about 20 to 30% per month. So this is the sample of 813 patients, and we can pretty much process data with a 2-week time lag. So anything that's happened in the preceding 2 weeks, we're able to sort of see and have completely processed. That was just their demographics, gender, and age. This is looking at BRAF status. So now for the first time ever, we're able to see what biomarker status looks like for patients across the country. This is information that hasn't been known before, but 88% of patients are tested with metastatic melanoma. It means that there's about 12% of individuals who have never been tested for BRAF status. And if those of you who don't know about melanoma, BRAF is one of the mutations for which we have a totally different set of drugs that we would prescribe in this particular scenario. Here is the BRAF mutation rates. So 42% of patients have mutated BRAF, which makes general sense based on what we'd seen historically in the literature, and BRAF mutation rates by age. So people who are younger have melanoma at younger age are more likely to be mutated. The third part of this then is actually trying to make sense of where patients are in their longitudinal story. In order to do that, we put together engineers and clinicians to ultimately develop what we would call lines of therapy algorithms or identification of treatment setting. One of the other pieces to this, and something we've been working on for a long time in CLHC, is that you actually have to think about analyzing and working with data differently. And we've been doing the same thing inside of Flatiron. So we have the software engineers sit at the same table as the statisticians and the epidemiologists and the clinicians, so that we can start thinking differently about how we work with it. And so in this particular case, what you see here are the patterns of drugs at an individual patient level that are being received across time. The diamond here in red is the metastatic date. And what we're trying to do is identify treatment setting where this patient first got PEG interferon, then was switched to vemurafenib, and then was switched to thalidomide and temozolomide. So these are 3 different treatment lines and 3 different points in the overall setting. And so what this allows us to do is understand where patients are in their journey and allocate that time frame to that point in the individual patient journey. This is important because in cancer, the way we think about people is, are we treating them before their disease became metastatic? We call that adjuvant. Or we treat them after the disease became metastatic? And if so, what point in the sequence? Is it the first drug you get, the second drug you get, the third drug? Because toxicity accumulates, and the way that you order these things is highly impactful. Putting all those things together then, a single data model, the ability to find true cohorts, the ability to have engineers and all the other people working with the data means that you can now start to accelerate understanding. So this is just some of our Tableau dashboards that we use to monitor what's going on, and I wanted to use this as a visual example. So we watch what happens across time, across the country, almost like you can imagine it becomes like the war room where you're sort of watching what's happening as the new data are coming out. And here what you see is this is January 2013, this is January 2015, and this big blue bar down here is a drug called ipilimumab or Yervoy, so a CTLA-4 antibody that came out in approximately 2012. This blue right here, the UNC blue, is vemurafenib. Vemurafenib is that BRAF inhibitor that I was telling you about. This gray right here is tafenilare mechanistic, and I'll explain what all this means in just a second, but I want to orient you. So that's tafenilare mechanistic. And this right here is Keytruda, and up there is Opdivo. All right, so what is it that you're really looking at? What you're looking at is the sequence of new technologies, innovations coming out in melanoma. So ipilimumab came out back here, and so did vemurafenib. Vemurafenib was the first BRAF inhibitor that we had. In October of 2013, GSK had approved by the FDA the next BRAF inhibitor, tafenilare mechanistic, and you see that blue, the UNC blue, start to be eroded by all of the gray. Meanwhile, ipilimumab was sort of doing its general thing until the PD-1, PD-L1 inhibitors started coming out in September. And you see the PD-1, PD-L1s come out. This was Merck's drug. It was approved by the FDA in September. Here's Opdivo. It was approved by the FDA on January 4th. It's that little white blip right there, and I can tell you the Opdivo coming out in the US. Oops, go backwards. But now, let's get smarter. Oh, just sort of drilling in. So Opdivo coming out. But what's the difference between when a person has their BRAF mutated versus their BRAF not mutated? Because one of the things we're learning in cancer is that cancer isn't just the buckets of breast cancer, colorectal cancer, and lung cancer anymore. Every single one of these diseases is being broken down into little bitty buckets based on the biomarkers and what we understand about the driver mutations in the disease. And so here, these patients, their BRAF is not mutated. So they should not be getting any BRAF inhibitors. And in fact, they're mostly getting ipilimumab, and now you can see that they're starting to get Keytruda, and this is the first line, so the first drug the doctors choose. This is the BRAF mutated patients. So these patients should be getting a BRAF inhibitor and not be getting any of these other drugs first. And what you see here is in the first line setting, in fact, they were mostly getting vemurafenib until Tafinlar Mekinist came out, and indeed that was exactly what happened. Here's the second line BRAF wild type setting. So this is now the second drug that the doctor and the patient choose together, and what you in wild type patients. And what you see here was ipilimumab was the dominant drug until all of a sudden in the second line setting, whomp, PD-1 inhibitors came into play. And what's interesting to see here is that oncologists across the country completely changing their practice. Not 14 to 17 years, but we're actually seeing these drugs come out really, really fast. And what we're also starting to see now is the impact of the adverse effects of these drugs as they're starting to come out. And this is the BRAF mutated patients. Meanwhile, one of the things I reminded us at the very beginning was the importance of getting information back. And so the last couple of minutes what I want to do is talk to you a little bit about getting information back on the practice side. So, right now on the research side of what we do, we spend a lot of time figuring out how do we create very efficient research mechanisms to be able to essentially do research at scale. But this all started by how we partnered with the clinical practice. And so in oncoanalytics and oncoEMR, one of the things we have is a big push to how do we develop tools that clinicians and patients and practice managers want. And the first thing that we learned in oncology was, lo and behold, they had no idea even what patients were coming into their practice, let alone how many breast cancer versus colorectal cancer patients that they had. So first we started teaching these practices some basic population management around who was coming into their practice. The next thing that we learned was that they would put staging data into their EHR if they actually knew which patients needed to be staged. So we started giving them essentially different kinds of information about the proportion of patients that, for example, needed staging data to go into the EHR. And then we also learned that there were a number of things that they wanted to have as almost like campaigns in the practice, like helping with survivorship care or palliative care. And so we started supporting with essentially campaign activities like survivorship care planning. Meanwhile, they told us, hey, we want to know quality metrics and quality monitoring, but we actually need to know more than just the number. I hit that measure 45% or 75% of the time. Help me understand how to make a difference. So in this particular case, this is GCSF or cytokine utilization for patients at risk for febrile neutropenia, where these are individual docs in the practice and what drugs that they're choosing for patients who are at moderate risk for developing febrile neutropenia and whether or not they're giving anything. And blue shows you when each individual doc gave no GCSF for a patient getting moderate risk chemotherapy. And orange tells you when they were getting a drug called pegfilgrastim. And what you see is high variability in one clinical practice across the usage of GCSF, even for a chemotherapy program with known moderate risk of febrile neutropenia. And so one of the things that we hear from these practices is don't just give us a number, start to segment it in ways that are meaningful to us. For example, we also show them for how much GCSF is being used when the risk of febrile neutropenia is almost zero. This is supporting discovery science. So now we can actually take all the same things and start to match it with biology. This is the picture, Reef, from CLHC. And these are our old patient journey maps where we had tissue collection. This is with Jeff Ginsburg's group, and the timing of when the tissue was collected, and then started to put that together, the genomic sample. And I use this as a reminder that we're now doing exactly the same thing. We know across the country where tissue collection has happened, where the tissue blocks are sitting, because we actually have that in the electronic health record data, and we can map that to the longitudinal clinical stories to be able to start to support, for example, biomarker development. And these are two things that we're doing right now. So Foundation Medicine We have a program where we are doing all the clinical annotation for Foundation Medicine's next-gen sequencing. And we also have just launched a program with the National Comprehensive Cancer Network, or NCCN, to rebuild all the outcomes databases for the 24 NCCN centers in the country. And so we're building essentially clinical annotation repositories for all those cancer centers that go along with their biorepositories, of which Duke is one of them. And the last piece then is also pulling it back to the EHR, because if you have one closed system, you actually can now put information back to the EHR. I don't have much to say about this yet because we just started doing it, but we're essentially building into electronic health record mechanisms to pull information, lessons learned back in real time. So the first thing is we realized if drugs are coming out that quickly, and you've got practices across the country taking up very complex drugs like PD-1, PD-L1 inhibitors, or CTLA-4 antibodies, give them real-time information that comes in about label-guided regimens, how to manage adverse effects, how to manage these drugs, and go ahead and do that. Present the information needed as opposed to letting them, making the practitioners go and find it. If you notice that they've actually been using this drug a lot, suppress that information. They don't need that information anymore, and don't put it in their way. So really starting to develop things in the electronic health record that go along with how doctors practice and what we're seeing, as opposed to just getting in the way. And similarly, since part of our mission is around improving research, one of the things that we saw was that the clinicians were using our Population Health Explorer to support clinical trials matching. They were actually using that same tool that we were developing for a survivorship clinic. They were using it for clinical trials matching. So now we're building the tools and technology to support clinical trials matching. So this is our new clinical trials matching tool. So for example, here what you see is we got 3 open trials in this particular practice. The system is continuously reviewing and identifying patients, a lot like we do at the DSR here at Duke. But then, as we identify patients and candidates for review and enrollment, what we're trying to do is think about how do we support the practice even better. So in this particular case, here's a lung cancer trial for patients who've got recurrent disease after platinum-based therapy. So we identify people who've received platinum-based therapy. We do pre-screening for different eligibility criteria, and we give a match confidence so you can prioritize the patients that you're looking for and that you're looking at in your practice based on how confident we are at the match. And further, we start to drill down on the individual inclusion and exclusion criteria so that you actually know what they passed on and also what they were likely to have passed on. And we can actually add other values as proxies in the EHR to start to signal for things. So for example, one of the eligibility criteria was one prior platinum platinum therapy, and there was a med order for carboplatin. So it means that this was likely met because we can find the administration order somewhere in the overarching system. And so really what we do is we start to identify and present to the recruitment coordinators how to think through that. So kind of bringing it back to Janet, we in Flatiron, and I think really in CLHC and certainly in my own head, always try and go back to like what it is that we're doing every day. How do we build learning health systems that take better care of real people? And if Janet was sitting on the couch next to you, what would she say was really important to do? Keep my information safe. Make sure that the work that you're doing is ultimately about me, so make sure that we personalize what we do. Figure out how to help my doctor get as fast as possible with all the rudimentary stuff so that he or she can spend as much time talking to me and really prioritize that as the #1 task. And also try and help accelerate the development of new solutions so that people like Janet don't die of diseases like melanoma. So with that, hopefully what we've done is gone from being turtles to watching new drugs coming out across the country literally by the month via linked patient level data, turning us into hares. So with that, I say thanks.
Thank you so much, Amy. That was fantastic. Impressive body of work. I want to really appreciate you came and special session today. Actually, want to thank everyone.
What?
Can't hear.
Oh, sorry. I want to thank everyone for coming today, and also just really wanted to appreciate that Amy made a special effort given her hectic schedule. So thank you for that.
Questions?
Can I, Amy, that was great and very inspiring. I wonder if I could, it seems like the core of this is the PDF review, and you kind of went over that a little quickly. I wonder if you could unpack that a little bit more. What is that process? How is it performed? It seems like there's an army of people somewhere reading charts the old-fashioned way. Is that?
Yeah, you know, so the question is, I moved quickly through the unstructured data review, and what's really going on there? And one of the questions people always ask me is like, is that really scalable, right? So first of all, actually, and at the time of the GO grant was this grant that we had here to do something very similar to this. We tried natural language processing. I spent time with IBM Watson. We've done a whole bunch of different things. And I was pretty convinced even before I joined Flatiron that really it required human review to do a lot of this work. So then the question is, how do you make it as efficient as possible? A lot of that is what I showed. How does it get actioned? We have literally an army. So right now we have probably 75 abstractors across the country, all of whom have to work a minimum of 20 hours per week and then go through almost like continuous learning and cycling and review. They are predominantly clinical tumor registrars, because there's a whole tumor registrar training that happens in the US, and so most of them were tumor registrars before, although about a third are nurses or PAs by background and pharmacists, and in fact the clinicians by background are by a lot better for the most part. What we find is that we call those little modules tasks. So imagine one CRF is comprised of maybe 20 tasks, so whether that's biomarkers, progression, et cetera. And on average, for example, a biomarker task takes about 5 minutes, whereas a stage task takes about 10, because it's a fairly complex thing to do. We have some tasks that are in R&D, so we haven't actually rolled them out yet. So for example, right now, we distinguish between visceral and non-visceral METs, but we actually want to get far more detailed about the site of MET and how the sites of METs are developing per person. So, because it's not just that you've got METs, it's actually that your METs accumulate across time, and so we're developing a set of tasks to try and both create a snapshot of current accumulation of METs and the time points that happened, as well as a mechanism to continuously evaluate that person as they continue to get their care. So then that brings me to the last piece, which is that every single time a patient's chart gets updated with any new document, it goes back to the abstractors for continuous review of the things that maybe have been missing. Does that make sense?
Yeah, sounds like a nightmare though.
Well, that's the reason why I gave you the example of that car, right? Like, because if you can imagine that the real task beyond the human abstraction is the technology to pull it all together and make sense of it. And so my war room is mostly, it looks like I'm managing a factory where like, it's exactly it. We have basically assembly lines, and you can kind of see what they look like. And over the course of each month, you can watch all of our tasks come in. You can watch us disperse them across the country and then come back in, and then the QA cycle and then in the data set. Sorry. Eric?
So thank you for coming. It is remarkable to see this taken from a cottage industry now to an industrial force, which is exactly what's happened. My question is just, you are building the data set that will be both in size and in scope richer and bigger than most anybody's. And once you reach that scale, there'll be large commercial value that'll be associated with it. But there's large potential value back to Janet. And the question I guess I'll have for you is, how will you deal with that potential conflict here? Because you've got science people all around this room who would love to use that data to write 6 abstracts or 10 grants or whatever, and you've got the FDA who would love this data to be able to track whether this new drug on market really is safe and effective, not use limited to claims information. So how does that work?
So the question is really, how do you maintain focus on the mission, recognizing that there's a commercial value to the overall data set as well. So we've tried to do several things. The first is that, number one is you actually have to set the culture that in order to have a company that's going to continue to serve Janet, it has to be a viable company. But we've tried to set also the culture that it's critical that we all understand why we do this. So, for example, we have actually inside of Flatiron we have knowledge infusion every week, but then one of those knowledge infusions a month is around patient care, so that everybody's sort of keeping focused on why they're doing it. Within the context of the overall network of data contributors, like Penn, for example, any academic organization that contributes data into the network ultimately has access both to their data in the processed format identified, as well as the overarching de-identified data set. So the idea being that essentially from the academic standpoint, accelerating use of the data and really trying to make it a clear part of what we do from the outset. So for example, at Penn, we have a committee that where we prioritize the kinds of data sets that they want, so that we're sort of packaging them as quickly as possible to get back to them. So that's like the second thing. The third thing is that we have intentionally tried to prioritize technology development of tools and solutions that ultimately we're going back to Janet. So that's one of the reasons why of all the things that we could be building right now, clinical trials matching was one of the things that we were doing, is because while many of the practices want to know, how do I make the most money on my drugs? We sort of said, well, one of the things that we can build first is sort of tools and technology solutions for direct patient care. So that's another thing that we did.
Eric.
Sorry.
Oh, me? So, Amy, wonderful talk. Thank you very much. You talked about data liquidity, and it seems like you guys have solved the data liquidity, at least internally, with a huge, basically mechanical Turk type operation. Now, with that in mind, what do you think about in terms of what has that experience told you about the ongoing efforts for interoperability, like FHIR and things like that? You say your bias is that a lot of these data are going to require a lot of human intervention and a lot of human curation. Yet on the other hand, everyone wants the ability to just exchange data without that much curation. So how do you resolve those tensions, and how do we move raise the level of the tide for all the rest of us struggling with other kinds of healthcare data?
So the question is really around this issue in context of data liquidity, and we're solving the problem by essentially forcing a forcing function to get everything onto one common data model. There are other things going on across the country, and there's an interesting. And Eric knows this, but fascinatingly, cancer has been the carve out, right? So all of the common data model activities are going on across the country. Cancer has been sort of off to the side. So, one of the things that we're trying to do is do the translation back to other data models. Right now we're working on OMOP. So we actually have tried to make the investment to say, how do we solve that internally to outside models that are happening? PCORNet is one that we're sort of like working on right now. Then I think across the overall healthcare community, the world of translation tables is going to have to get more and more commonplace. I also think that we need to ask ourselves at sometimes, like, what's the real purpose and what are we trying to get to? And a lot of the activities that have been going on within the context of data liquidity to date have not necessarily always understood the end game in mind, and I think that that's going to be one of the tasks there. I used to be part of the Data Liquidity Coalition, and I left it. And the reason that I left it was that it was a lot of hot air, and actually people didn't want to do the real work of like, this matches this, and here's how we think through it. That help? I'm not sure it solved it.
Okay, that was terrific. I want to follow up from Dan's question a little bit more and ask you whether you see these human abstractors, even with technology-enabled tools, is an intermediate step. And ultimately, whether natural language processing and other purely algorithmic solutions are going to automate this process. And if so, what are the principal barriers at this moment to achieving that?
So the question is whether or not really artificial intelligence and machine learning will ultimately do the task for us. Did you ever see War Games? You know, Whopper, the Whopper. So, you know, like I think that one of the things that Whopper always reminds me is that for those of you who didn't see War Games, like there was the belief from the professor that the Whopper was going to actually ultimately be able to solve our understanding of how we defend our borders from a military strike and know when to set the missiles off. And in essence, one of the things that happened was in the end, human intervention became, still fundamentally critical. And I think that the same thing is going to happen here. Like I came from an AI background, and I always believed, even in my old AI world, that there's a point beyond which, at least with current ways of thinking for artificial intelligence, we still need human interpretation to be able to pull together the story. And one of the things that we've seen in natural language processing is that it works well and better in different kinds of problems. So outside of cancer, many problems actually can be solved well with natural language processing and/or other machine learning and artificial intelligence approaches. And your background's in radiology, and certainly radiology is one of the places where there's been incredible strides made. And we've seen the same in heart failure and COPD, for example. So I think it's actually going to be spotty and different by diseases. Diseases that require complex interpretation is the place where I don't really see ultimately computerized techniques solving the problem, but I do think that they're going to solve a lot of the problems. So the intermediate step for what I've just showed you is biomarkers. Some of that's ultimately just going to be digitized data, and some of that should be able to be done by natural language processing. I can show you an entire slide deck right now why we can't do biomarkers by NLP at this point in time. It's actually quite fascinating. So that's a good example of what should happen that way. The metastatic progression that I talked about, I think that should be done by probably an algorithm within the next 2 years. And what we do is we have our software engineers build algorithms in line with our abstractors and see if we can't, get them. They're basically in competition. It's like a leaderboard.
Great, we're out of time.
Thanks,
I really want
guys.
to thank Amy again for a wonderful presentation.
Thank you.